let's take a look at the hybridization experiment in a picture. So here Mendel is crossing two plants, a purple flowered plant and a white flowered plant. Here you can see that the first generation, all of the offspring are purple. Therefore, we can determine that purple is dominant to white. White is recessive to purple. And what is noticed here is that the entire F1 generation, the first filial generation, is purple. Now, the P2, or the second parental generation here, the P2 shows us that that white recessive trait pops back up. So when we cross two purple flowers from the F1 generation, we get a 3-to-1 ratio, 3 purple flowers for every 1 white flower. And this is Mendel's classic hybridization experiment. We can put some symbolism to that, and we can put some numbers to it. So notice in the F1 generation, as we are combining these two, when we're doing our P2, our second parental cross now, half of these alleles are going to separate into gametes here. Half of these alleles will separate into the gametes there. So the gametes will be half dominant and half recessive. Okay, we can include that in the Punnett square. And it develops as you can see here, a 3-to-1 ratio. 3 of these plants will be dominant, either homozygous dominant or heterozygous. And 1 of these plants will be homozygous recessive. Now, Mendel had some good fortune, certainly. The fact that all 7 of these things were simply dominant. The fact that he inherited true breeding lines. But where Mendel came to his own was the fact that he collected large samples. He had meticulous record keeping. He had some hard work to go along with his good fortune. Here are some of Mendel's actual data. You can see the number of offspring that he had produced and counted. Notice the ratios all come up very close to a 3-to-1. Mendel's model that he developed had to do several things. First of all, it must refute the common thinking of at the time that the offspring were merely a blending of the 2 parents, that a tall plant and a short plant would produce a medium height plant. And certainly we know that is not true. Okay? If that were true, if that's how inheritance was passed on, certainly every plant and every organism in a population would over time begin to look very much the same. Okay? But that was commonly thought to be true and had to be refuted. So also these heritable factors, or these particles, needed to be shown to be able to be carried, need to be shown to be able to do the things that Mendel saw happening in his offspring. So the first thing he developed was the law of segregation, in that when these inheritable factors, which we would now call genes or alleles, these inheritable factors did in fact separate during the process of meiosis. All of this is pretty remarkable if you consider that Mendel probably had little or no knowledge of meiosis, probably had little or no knowledge of those kinds of cell division processes. Certainly had no knowledge of DNA or anything of that nature. So the fact that he figured this out from its foundational level is pretty amazing, really. So now we've got 2 factors for each trait. They separate in meiosis, and you get 1 of those from each parent. This explained